Hi, so I'm Stephanie Booth, and I'm based at the Canadian Science Centre for Human and Animal Health in Winnipeg. This is a facility dedicated to diagnosis and surveillance and research on infections of human and animal diseases. So in our lab, we're hoping to generate more data on the phenotypes of the cases that we see in Canada and the strains and the subtypes, MM1, VV1, and we're hoping to be able to determine more atypical cases and investigate those in more detail, especially given the rise of CWD in North America and how it's expanding the range of disease in deer and elk across the country. And we'd like to be able to keep a closer eye on the types of CJD that found. So to provide a focus for this retrospective study, we decided to use tissues from cases originating from Alberta and Saskatchewan. Given that these provinces are the epicenter of the CWD epidemic in Canadian deer and elk, we decided to assess the heterogeneity of CJD cases that already exist in these provinces as a baseline for enhanced surveillance. And we started with 32 cases for which we had frozen tissues available in the lab. These cases were of various subtypes as listed in this table. And we also had information available such as medical histories, including any history of neurosurgery, and we have some brief description or history on whether these people participated in hunting or whether they'd consumed cervid meat at any time. So in the first instance, we decided to develop an assay that we could use to screen multiple samples simultaneously to assess for heterogeneity in the conformation of CJD prions. We hypothesized that variations in the prion conformation are intrinsic to the strain characteristics of those prions and ultimately to the disease phenotypes, and that these may be detectable by methods that measure refolding kinetics. So we therefore investigated whether the seed substrate pairing has a direct relationship to the conversion rate using the quick assay. So this slide provides a quick primer to what's happening in the quick assay, which you're probably fairly familiar with, but briefly, we add misfolded prions from a CJD brain homogenate, and this induces the refolding of the normal prion protein to produce aggregates. And using vigorous shaking, we break up these aggregates to amplify the number of seeds, and this can be repeated cyclically as a chain reaction. And usefully, the aggregated protein is detected with incorporation of a fluorescent dye. And so we hypothesized that when we have conformationally distinct CJD, they replicate with slightly different kinetics in multiple different substrates, so producing a kind of fingerprint for each type or subtype. So what we did was prepared a number of different substrates using full length and also truncated prion genes from different species. And so we measure the conversion rate by comparing the time at which fluorescence in a given reaction exceeds the predefined threshold, which we call the lag phase. And we correlated each plate using a standard on the run. So each run we use an identical prion sample and we can normalize each plate quite accurately, we found, with this method. And so in this first graph, we show how the lag phases in these three different substrates compare with, I think in this graph, there's about 25 cases on there in a bank vole, hamster, and a mouse protein substrate. So we found that this assay was rapid and scalable, and it enabled the direct comparison of these many different cases, as you can see. And we found that we have one major cluster and we have some outliers that I've colored in. So we see these outliers at the bottom of the graph and at this end of the graph. And when we correlated these outliers by looking at the biochemical analysis on a gel and by comparing with the clinical and pathology reports for these cases, we found that each of them had some atypical feature related to either their pathology or their biochemical Western blots. So this blue case, for example, has slightly higher bands that we see on the Western, particularly the diglycosylated isoform. The pink sample colored here had a 20 kilodalton band that ran in between 2119, the typical type 1 and type 2 case molecular weights by Western blot. And this sample annotated in turquoise, it looked identical to a typical one case on Western blot. Here the one cases are colored gray, the V2 cases are colored in brown, but it looked identical to a typical one case on Western blot. But when we did a denaturation assay, we found that it's had different properties. It seemed to be more stable by temperature. We can see here on the graph and on the plot, and we can see these 3 denaturation profiles for 3 different MM1 cases, and this was our atypical one that seemed to be more stable. So out of 18 or so MM1 cases that were all identical, this one did appear to be slightly different. So we do think that outliers are showing cases with atypical features quite accurately. Interestingly, these 2 green cases that we found were also looked very atypical on our diagnostic surveillance Western blots, although type case A was designated as a VV type 1 case and case B was designated as a VV type 2 case through surveillance. But when we ran them on a Western, we were suspicious based on their long duration of disease, 45 months for one of the cases, 14 for the other. One of them did show remarkable on the pathology. Nothing abnormal was or remarkable was highlighted, but we were suspicious that they might be VP variably protease sensitive cases, and so we ran them on a Western blot with antibody 1E4, and we did see these typical bands that confirmed they were VPSPR profile cases. The next assay we started to develop was a more sensitive Western blot assay using a simple Western capillary-based capillary immunoassay. And so we decided to look at this system. One of the main reasons is its increased sensitivity over normal Western blots and its ability to perform semi-quantitative analysis. So here we used recombinant hamster protein just to look at the sensitivity, and we found that it was at least 10 to 100-fold higher than traditional Western blot using the antibody 3F4. And this is the output you get for the capillary-based electrophoresis. It can produce a phony Western blot picture, or the main output is this electropherogram, and we can quantitate the band equivalent to the band on a gel by calculating the area under this peak. So next we tried loading proteinase K digested protein isolated from a human CJD case. We prepared the protein from a 10% brain homogenate of a subtype case, used proteinase K to digest it, just like we would do for the Western blot. This is, in fact, the Western blot in panel A, and then when we loaded it onto the capillary, we basically just get this big messy peak, as we see in B. We suspected it contained high molecular weight aggregates and insoluble aggregates, which cause this peak, even though it looks beautiful by Western. And when we increase the molecular weight analysis, we do see that there's a lot of insoluble high molecular weight aggregate on the electropherogram. After this messy band wasn't really useful at all, and we did try many different methods to kind of improve the results that we saw. We loaded different amounts of protein, we loaded different amounts of PK digestion, we used different buffers for loading, we used different amounts of starting material, and nothing made any difference to this. We always got this big messy peak. We were almost about to give up, but then we decided to try phosphotungstate precipitation, which is a method that's often used to precipitate and concentrate infectious prion protein from tissue samples. So we used PTA precipitation prior to digesting, and this is what it looked like on the gel. It's the same starting sample that we used in the Western in A. The Western profile looks a little bit different, but when we loaded this protein onto the capillary, we saw a huge difference. We're immediately able to see our 3 glycoforms bands, the unglycosylated, monoglycosylated and diglycosylated band. When we ran it out to look at high molecular weight, we did see a tiny little bit of aggregation, but basically all this big aggregated mess of protein disappeared. After PTA precipitation, we did use type 1 and type 2 proteins on the capillary, and in each case we used about 40 samples, and in each case we were able to discriminate type 1 or type 2 based on the electropherogram molecular weights. And particularly useful was the use of a lower matrix, a matrix that is dedicated to looking at tiny proteins, 2 to 40 kilodalton proteins. And when we load our prion protein onto this matrix, we only see the unglycosylated and the diglycosylated band, but the assay was even more sensitive, and gave us much more confidence in diagnosing type 1 and type 2 by the electropherogram. So we do have a number of cases that appear to contain mixtures of type 1 and type 2 prions in our cohort. This is a Western blot of one of those cases. In panel A, it clearly shows the 2 bands at 21 and 19 kilodaltons. But after we PTA precipitated, we always got this single banding pattern. We thought this was unusual, and we actually picked out quite a number of different cases where we had these mixtures, and we always ended up with a single banding pattern after PTA precipitation. And when we run these on an electropherogram in panel B, we always get these nice peaks, no hint of a double band. So we went on from this to actually take PTA precipitated type 1 and type 2 proteins. We mixed those together, and then we loaded onto the electropherogram onto the capillary, and the electropherogram showed again a single band, but there was always a slightly wider band for the unglycosylated protein than the sharp peak that we see before. And so our conclusion was that if we have a mixture of bands, we can't discriminate as closely as 2 kilodaltons by the capillary analysis method. So given that the capillary was very sensitive for small molecular weight proteins on the matrix that was specially designed for 2 to 40 kilodaltons, we decided to look at the variably protease sensitive cases. Here we show the Western blot for those cases with antibody 1E4. I didn't show 3F4 because the bands were so faint. One of the cases was in panel B, one in panel C, and those were run with 3F4, and we clearly see these small bands with 3F4. E and F are the same cases run with antibody 1E4, which clearly shows similar pattern. This just shows the increased sensitivity of the capillary method, that we could use 3F4 with a lot of confidence in this case. And we actually can look at these 2 cases together by combining the electropherograms, and we can see we can look for subtle differences in the banding patterns of the tiny proteinase K resistant bands with quite a lot of confidence in discriminating the different sizes using the capillary. So we think it's particularly interesting or particularly useful technique for looking at variably protease sensitive cases and looking for small molecular weight bands. So next, we've just started looking at a new animal model to analyze our CJD cases. So we do have a colony of deer mice that our lab holds for various other infectious diseases. And so we decided to inoculate them with CJD and see what happened. And we did find 100% attack rate for sporadic CJD of type 1 in these deer mice with a DPI of about 230 days. And this decreased to about 160 days on the second passage. We got really high titers of prions, so this seems to be a nice model for CJD. And as I mentioned, we're just at the early stages. We've passaged a couple of the atypical cases, and 2 of these, they do seem to have different incubation periods after we've inoculated them from the typical, which was about 230 days post-infection, but we're still at early stages and we haven't looked at all these tissues so far. We have also inoculated with chronic wasting disease from elk, and this gave an incubation period of about 400 days on the first passage. And we've also just started inoculations with V2, MM2, and some further atypical cases. So we're trying to develop this model and see how it goes. So that'll be something for the future. The animal model has been a little bit delayed due to COVID. So I'd just like to give some acknowledgements. Thank you very much.